Assalamu alaikum guys, welcome back to another video by Science with Kasmani. My name is Sanjanin and today we are gonna be having a P5 masterclass. Basically everything which you need to know about your P5 exam or before your P5 exam in fact. We'll cover all of that today. Plus at the end of everything we have covered, I'll solve a past paper, a complete past paper with you guys so that we're able to apply all of the information and all of the knowledge which we just learned into that specific past paper. So be sure to watch this video right until the end and I can guarantee you're going to be prepared for your P5 exam. All you need to do after completing this video is just practice two or three more papers and that's it. You're more than good for your exam. So I've written everything down over here. We're going to be looking at each of the pointers that I have noted down one by one. So let's start. The first thing about P5, let's start from the very beginning that you need to understand is this. So this right here is a table from your syllabus file. And it says that when we talk about the P5 exam, it consists of two parts, wherein the first part is the planning part, and the second part is analysis, conclusions, and evaluation. In your exam, you will be tested on four skills: how well you are able to plan the experiment and essentially plan your answers, how well you are able to analyze the given situation at hand, how you're able to conclude conclusions from certain experiment, and how you're able to evaluate your own answers. These are the four skills that you're going to be tested on. So, planning is one of the skills that is for 14 to 16 marks, whereas analysis, conclusions, and evaluation together is a skill which can get you a maximum of 14 to 16 marks in the exam. All right. Now, when we talk about paper 5, I personally believe that paper 5 is divided into two parts. Now, I'm not talking about the division of questions that, let's say, there's a question one, there's a question two. I'm not talking about that. I'm talking about the content in the paper. When you're studying the content in the paper, what's going to come in the paper, you can divide that content into two parts. The first part being your own knowledge from the syllabus that you have learned in P4. This is going to be the first part of what you're going to revise together. And we're going to revise this together today as well. And the second part of this paper is going to be about basic statistics. So for those who take A level math as well, this is going to be a bit more easier. Even for those who don't take maths, but I didn't take math in my A levels, I still got through. So it's not that tough. Even if you don't take maths, it's basic statistical tests and basic statistics. In biology, we call these biostatistics. So if you look at the word biostatistics, it's biology and then statistics along with biology. So together combined, that brings about the second part of the content in total that you need to know about. Right. So let's talk about the first part and then we'll move on to the second part. So basically, right now, what we're going to cover is the knowledge part. How do you know what part of the P4 syllabus you're going to have to revise for this exam again? So let me tell you, the first chapter, or you could say the only chapter which you have to revise is chapter 18. Now, if you don't know what chapter 18 is, this is the biodiversity and classification chapter. No problems at all. If you don't have the time to revise this, we're gonna do that. That's what this video is about. We're gonna revise every part of this chapter together. So, biodiversity and classification chapter, you're gonna have to revise. And then after that, you're gonna also have to remember how some other experiments, those experiments not mentioned in chapter 18, how some of those other experiments work. Now, you might have gone through this in a random question in a P4 or a P2 in AS levels, or you might have just looked at this in a random diagram any point in these 2 years, some random experiments throughout the 2 years in your A levels, you also have to remember. And we look at what these other random experiments are. So, let's first talk about this biodiversity and classification. I'm going to first list down all of the topics that we're going to cover, and then we'll start looking at each one of these in detail. So, number one, Simpson's Index of Biodiversity. Number two, the chi-squared test. Then, number three, the t-test. Number four being the Lincoln's Index, which some students recognize this as the mark release recapture method. So let me write that two in the bracket. Mark release and recapture method. Okay. And then there is a number five as well, which is the Hardy Weinberg equation. Then number six, this is Pearson's linear correlation coefficient. And then we have the last but not the least, this is the Spearman's rank correlation. So basically the two correlation coefficients at the end, Pearson's correlation and then Spearman's rank correlation coefficient. Let me just double check if I have mentioned everything and I have not missed out on anything. So yes, everything else is done. All right. So let's talk about each one of them and discuss each one of them in detail. First comes up Simpson's Index of Diversity or Simpson's Index of Biodiversity. What is this? This is basically a statistical test which is used in order to measure species diversity. It is the statistical test which is used to measure species diversity. Now, what is species diversity? Let's try to understand this. Species diversity is basically the different types of species as well as their relative abundance within an ecosystem. So we're first counting how many species there are. Are there four species, five species, six species, seven or eight species, etc., etc. And then we are also counting how many organisms are in each specific species. So species and a combination of those two is also known as species diversity. So species diversity is just a measurement of the number of species and the relative abundance. Are there 40 organisms per species? 50, 60, 200, 500? What is the number of organisms belonging to a particular species? So, and their relative abundance. And where are we measuring this? We are measuring this by looking at a confined ecosystem, by looking at an isolated ecosystem. So it is what does an isolated ecosystem mean? No organism can go out of the ecosystem, no organism can come into the ecosystem. So it is a measurement of the number of species and their relative abundance within the ecosystem. Why am I repeating this definition again and again? Because this comes up as a two marker in your P5s as well. Define species diversity. So just make sure you remember this. Now, there is also another word you can use to define number of species and also another word you can use to define the relative abundance of each species in the ecosystem. So for this, you can use the word species richness, and for this, you can use the word species evenness. If you write what I've written down in orange, it's fine. If you write these words, it's also fine in the definition of species diversity. How do we measure species diversity? Let's understand the method behind it. So I've got an example for you guys, and we're going to discuss it together. So let's say I have four species. I'm going to write the column heading as type of species. So it's basically is four different types. The first type of species is called species A, then there is species B, then there is species C, and then there is species D. Now, what I've written in the second column is the letter N. That's the column heading, right? What does N mean? N here refers to the number of organisms. So let's say you walk into an ecosystem and you are measuring how many organisms are of a particular species. And you find out for species A, let's say you have 20 organisms. For species B, you have 25 organisms. For species C, you have 22 organisms. And for species D, you have 18 organisms. Now take a look at this. Capital N, where small n refers to number of organisms, capital N, you guessed it, it if you, even if you didn't, it's fine. It refers to total number of organisms. So how are we gonna get the total number of organisms? Basic stuff. Just add all of the numbers together. So this is gonna be 20 plus 25 plus 22 plus 18, which is equal to 85 organisms in total. Now, what I'm gonna have you do is pick up a calculator. For each species, take their number of organisms in the numerator and the total number of organisms of all four species combined in the denominator. So basically, n, small n, divided by capital N. Let's do that for each one of these. So for the first one, it's gonna be 20 divided by 85. For the second one, I'm gonna write the answers later. For second one, it's gonna be 25 over 85. Then for the third one, it's 22 over 85. So that's their simplified fraction. And then for the last one, it is 18 over 85. If I put all of these numbers to three decimal places, or you could say three significant figures, this comes out to be 0.235. This comes out to be 0.294. Then this comes out to be 0.259. And then the last one is 0.212. All right. Now, next step. All of these values which you got by dividing number of organisms divided by total, square all of these values. So now what you have to do is n divided by capital N, what you did before, but just take the square of each value. So meaning 0.235, put that in the calculator and then square it. So this comes out to be to three decimal places, 0.055. Make sure all of your significant figures, if you're looking at significant figures, they're consistent. If you're looking in terms of decimal places, make sure they are consistent as well. Meaning your first answer should not be to three and second answer should be to one decimal place. Make sure it's all to the same number of decimal places. Then I'm just going to write this quickly. It comes out to be 0.086, then 0.067, and then 0.045. All right. Step number 4. Find the total of all of these values. And this will make sense in a second. Meaning, you need to find the sigma, the total. This is the sign refers to the word sigma, which in English refers to the word total. So what is the total of small n over capital N whole squared? It's just the sum of all of these values. So 0.055 plus 0.086 plus 0.067 plus 0.045. And this comes out to be 0.253. That's the sigma n over capital N whole squared. Last step. Takes the number 1 and subtract it from this value. So can we say the formula is going to be 1 minus sigma n over capital N whole squared? Yes, we can say that. And eventually, the answer is just 1 minus 0.253, which comes out to be 0.747. This right here is the value that we are looking for. So let's do a summary of all of the steps which we solved or all of the steps that we performed. Step number one, find out n for each species. What was n? Number of organisms. You need to find out how many number of organisms belong to a particular species in each ecosystem. Or if you're looking at one ecosystem, right? So in one ecosystem, how many are species A? How many organisms are in B? How many in C? How many in D? That's finding out small n for every species. Step number two, find out capital N, meaning the total number of organisms in that ecosystem combined together. That's capital N. Step number three, find out small n divided by capital N for each species. And then the last step is, the second last step is find out small n over capital N squared for each species. And now the final step, just when you're done with that, find out sigma small n over capital N whole squared. That's it. Now you can apply the Simpson's index of biodiversity equation. What is the Simpson's index of biodiversity? 1 minus sigma n over capital N whole squared is the Simpson's index of biodiversity. Now, here's the good news. In your exam, you're not going to be calculating all of this. Not at all. You might have also seen this when you're solving some past papers, that the examiner or the question has already done this, this. Of course, you can't calculate them, you can't count the organisms, right, from a paper. You have to physically be there in that ecosystem. So the examiner has done this for you, this, and this sometimes as well. You just have to subtract this value by one, and on top of that, even the formula is given to you. So you pretty much don't have to do anything. That question is just testing if you know how to use a calculator or not. Simple as that. It's 1 minus sigma small n over capital N whole square. Now, what you have to understand here is what is the significance of this value right here. So, when we talk about the values or the results of the Simpson's index of biodiversity, these results are represented by the letter capital D, D for diversity. Now, the Simpson's index of biodiversity values can be less than or equal to 1, and it can be more than or equal to 0. Now, here's the important part, and I'm writing this in the form of a note. The closer the values are, the closer the value which you get after doing all of these 6 steps to 1, the closer your value is to 1, the higher the species diversity. Meaning, in an ecosystem, it's going to be more number of species, that species richness, and every species is going to have too many organisms. That species evenness. So, species richness is going to be higher, species evenness is going to be higher if the species diversity is higher. How do you know if the species diversity is higher? If the value is closer to 1. So the higher the species diversity. So when you know this, you can now say that, oh, the species richness is going to be higher. And on top of that, the species evenness is going to be higher. And there is one more thing as well. And that is you now have a more stable ecosystem. Let's understand this point, and this is really, really important. Okay, let's take an example. Let's say we have a predator in this ecosystem. Let's say you just drop some lions or you put some lions into this ecosystem, 20 or 30 lions. Turns out these lions like the color purple. Now, let me ask you this. Compare two ecosystems, right? In one ecosystem, you have lots of different species, and each species has lots of different organisms, and you have different phenotypes, meaning different colors. Colors is the phenotype in this case. You have purple organisms, black, blue, green, yellow, red, and pink. Versus an ecosystem which has just two species. Each species has 2,000 organisms, so 4,000 organisms in total. And out of these 4,000, 3,000 are purple. Let me just write down. You have an ecosystem 1, let's call this E1, and you have ecosystem 2, let's call this E2. Now, E1 has many species, and with many species, it has many different colors of organisms. Can we say it has a high species diversity? Yes, we can. Their Simpson's index value is going to be closer to 1. So it's basically many different colors. Versus in ecosystem 2, it has a very low Simpson's index value. It's more closer to 0 than it is closer to 1. And it means that the species diversity is going to be low. The organisms are going to have a limited number of phenotypes. Let's say most of these are going to be pink. Or if I can find the purple color, let's see. Yeah, purple as well. Now, think about those lions. Think about that selection pressure, those predators. They like the color purple. If they like the color purple, which ecosystem is gonna suffer more? It's gonna be ecosystem 2 because it has more purple organisms. It has a lower species diversity. Compared to number 1, only a few organisms are gonna die off. Only a few organisms are gonna be disfavored by the selection pressure. So you see, at the end, after the lion has done its job, these are the remaining organisms in species ecosystem 2, and these are the remaining organisms in ecosystem 1. So, which ecosystem is more stable? You would say ecosystem 1 is more stable. Why is ecosystem 1 more stable? It's because it had a greater species diversity. It was able to handle the selection pressures much better. So, the greater the species diversity is, the more stable your ecosystem is, or in other words, it can handle the selection pressures much better. That is all that you need to understand. All right. So, we talked about the Simpson's index of biodiversity. Let me just put a tick next to that. So, chi-squared test. This is basically a statistical test which you must have learned in chapter 16 when you were studying inheritance, monohybrid inheritance, dihybrid inheritance, blah blah blah. So that's basically chi-square test where you must have learned that. So what is chi-square test? Chi-square test is a test, obviously, that is used to check if there is any significance between observed and expected numbers. Chi-square test is a test which is used to check if there is any significant difference between the observed and the expected numbers or not. Now, if I was in your position, I absolutely have no idea on what this statement means. So let's take an example. Let's say we have a gene, and just assuming, please don't memorize this example. I have a gene, and this is gene A, or let's call this gene 1. That's the name of the gene. Now, this gene 1 is actually coding for body color in a species of rats. So it's a rat species wherein this gene called gene 1 codes for body color. Now, there are two alleles of this gene. There is a capital A allele and then there's a small a allele. capital A, of course, is written in capital, it's the dominant allele. And when this dominant allele is expressed, the phenotype is black color. And when the recessive allele small a is expressed, the phenotype is blue color. In other words, black is the dominant trait and A is the recessive trait. Let's say I take a homozygous dominant. I take a homozygous dominant organism, meaning that both of the alleles in the genotype are going to be dominant. That's capital A, and I mate it with a homozygous recessive, which obviously means their genotype is gonna be small a, small a. Obviously, whenever two organisms are being mated together, it's not their genotype which combines together, right? It's their gametes which combine together. Now, if you look at their genotypes, these are basically two sets of chromosomes, right? And this is why this is a diploid cell, which is why we call it 2n. But in this case, so this is also diploid, which means 2n. But when their gametes are going to be formed, remember, gametes are sex cells. These are reproductive cells that are haploid in nature, meaning they have only one set of chromosomes, which is why we call them or we represent them by the letter N, this letter right here. So if capital A is two sets of chromosomes, you can only pass down one set of these chromosomes. So either this capital A passes downwards or this capital A passes downwards. Either way, it's the same thing, right? It's a capital A being passed downwards. So this is the gamete here and this is the gamete here. Now, these two gametes fuse together in order to form a zygote, which is basically fertilization. And there we have it, the organisms, the offspring's genotype, in fact, is capital A, small a. Can we call this the F1 generation? Of course we can, because F1 generation stands for first filial generation. It is the generation of offsprings that is formed after the first mating process. So basically, this diagram here is telling us if let's say you have 20 offsprings produced in this F1 generation, each offspring is gonna have the same genotype, capital A, small a. Now let's say I take this F1 generation, meaning not F1 generation, I take one of the offsprings from F1 and then I take another offspring from F1. So it's basically a capital A small a and then a capital A small a. And I mate them together. What's going to be produced? F2 generation. Now, what I produce is the F2 generation. But the thing is, I produce 600 organisms in F2. So this is just my example. We're producing 600 organisms in the F2 generation. Let's try to figure out what the genotypes and the phenotypes of the offsprings in the F2 generation is going to be. So, if it is capital A, small a, only one set of chromosomes can be passed downwards, meaning there are two possibilities this time. Either the gamete can be capital A or the gamete can be small a, and same case here because this is also heterozygous, this can be capital A gamete or this can also be a small a gamete. Meaning, when we now talk about the offspring's genotype, it can be capital A, or it can be capital A small a, or it can be capital A small a, or it can be small a small a. These are the four genotypes. Both capital, that's homozygous dominant. Then two of these are heterozygous, and then one is homozygous recessive. Let's try to write down the phenotypes for these. So this is gonna be, what's the dominant trait? Black. So this is gonna be a black color. Then this is also black. The third one, which is also heterozygous, is also black. And the last one is blue. So it's just two phenotypes, right? Either black or blue. Turns out that the black organisms out of 600 are 405. And the blue organisms out of 600 are 195. Let's try to double check if they add up to 600. Yes, they do add up to 600. So black are 405 and blue is 195. These numbers right here are your observed numbers, isn't it? Because how did you figure out which phenotype is which? By looking, by observing. You observed that out of 600, 405 are black, and out of 600, 195 are blue. But were the 600 organisms actually supposed to be distributed in this ratio? Let's take a look. Now, how do we know that? Our question right now is, how are we so sure that the 600 organisms were actually meant to be distributed in this way? Simple. Let's do a Punnett cross. Capital A, capital A, small a. Basically, just do this again. It's the same thing. So I have one homozygous dominant, I have two heterozygous, and I have one homozygous recessive. Meaning, can we say that there is a 75% chance that the dominant allele is expressed, right? It is expressed here, here, and here. Three out of four cases, it is expressed. So 75%, the dominant allele is expressed, and the phenotype is black. And there's only a 25% chance, one out of every four offsprings are gonna be blue. Let me ask you this. What is 75% of 600? Let's try to calculate it. 75% of 600 is 450. And 25% of 600 is obviously 150. 405 and 195 were your observed numbers, the numbers which you actually got. But 450 and 150 were supposed to be your expected numbers. Now, here comes our topic. Chi-square test. Now read this again. It is used to check if there is any significance between the observed and the expected numbers. Now you're checking, is this difference big or is this difference small? Is the difference between 405 and 450 big or small? And is the difference between 195 and 150 big or small? Now, the question which I get asked most is, why do we use a chi-square test for this? Why can't we just say this ourselves that, oh, 450 and 405, this is a big difference, and this is also a big difference. There's a 45 organisms difference, right? Why do we need to do a test for that? It's just the way this works. Doing a statistical test is a good option in order to prove something, because some people can say that this difference is large, but other people can say that this difference is small, right? It's the way people look at colors. If I look at this phone color, I can say this is orange. Someone right here who has a different amount of light falling on their eyes can say this is pink. It's different opinions, and this is a very subjective matter. Some can say this is a very big difference, some can say this is a very small difference. So what do? We just perform a statistical test to prove if it is a big difference or if it is not a big difference. And this is where the chi-square test comes in. So how do we perform the chi-square test? I'm going to write down all of the steps and then we're going to do these steps together. So step number one, make a null hypothesis. Now, many students find this really tough, but actually, this is really simple. The word null means of no significance. You must have heard the word null somewhere before, right? Null as in zero or no significance or nothing. That's just the word null. Now, we have all looked at what's a hypothesis. It's basically something that scientists assume before looking at an experiment. But what's a null hypothesis? It is a hypothesis of no significance, of no difference. So the null hypothesis here is you're gonna say that there is no significant difference between the observed and expected numbers. Every single time the chi-square test comes up, this is gonna be the same sentence which you write down. There is no significant difference between the observed and the expected values. Now note I have written the observed values as O and the expected values as E. So that shouldn't really be a problem. So there is no significant difference between observed and expected values. Now you've already made a null hypothesis. Step number two, calculate the chi-squared value. Now remember Simpson's index of diversity? It was denoted by the letter D. Chi-square test, the word chi is denoted by x squared. So this is basically stands for chi squared. So calculate the chi-squared value. How do we calculate that? So first, I'm going to write down the two phenotypes. That was black. Let's write the dominant one first, and then blue. If you look at the observed numbers, black were 405, blue were 195. But if you look at the expected numbers, black were 450. It was supposed to be, in fact, 450, and blue was supposed to be 150. Now you have to write down the observed minus expected. Step number one. Take the observed, take the expected, subtract them together, write your answer along with the sign. So 405 minus 450, let's do double check. This comes out to be as minus 45. Don't worry if it's a negative sign. 195 minus 150 is going to be 45. There's no use of writing plus 45, so it's just 45. Step number two, square the values which you just got. So observed minus expected, just square it. So what is minus 45 squared? Remember, put the minus in the bracket. Never do this basic amateur rookie mistake. Don't do this. Do this. Put the minus inside of the bracket. You should know this by now, guys. So minus 45 whole squared. This turns out to be 2025, and then 45 whole squared is obviously the same number, that is 2025. Step number three, take these values, so observed minus expected squared, and then subtract it, sorry, divide it by the expected value. So 2025 divided by this expected, because we're looking at this column for now. So divided by 450, that is 4.5. And then 2025 divided by the expected for blue this time, that is 150. So this is 13.5. Now, the last step, just like in Simpson's index of diversity, not the one minus, the one before that, take the sigma of observed minus expected squared divided by expected. And this comes out to be 13.5 plus 4.5, which in this case is going to be 18. This is your chi-squared value, which is 18. Now, step number three, which is determine the degrees of freedom. Now, this right here is a very controversial topic in the sense that 90% of those students attempting a P5 paper don't understand what the word degrees of freedom means. Now, I've posted a reel on this too. You can watch that, or I'm just going to explain the same thing as I did in the reel. So, listen to this. What does degrees of freedom mean? Let's say I have four values, right? I'm not talking about chi-squared anymore. I'm just explaining degrees of freedom. Let's say I have four values, and the total of these four values should be equal to 30. So I can pick any values I want, but the only catch is it should be equal to 30 when I add all of these together. So let's say I take the first value as 7, I don't know, take the second value as 9, take the third value as 3. 7 plus 9 is 16, 16 plus 3 is 19. Do I now have any control over the fourth value I choose? Can I just write 1 here? I can't. The fourth value, choosing that is not in my hands. I just have to write the fourth value as such that the total comes up to 30. So this is 16 plus 3 is 19. And now forcibly, meaning I'm not doing this on my own, I have to write down 11 here. How many values did I have a control over wherein I could write anything? Only three values. Out of how many? out of four values. This is the exact same concept here. Degrees of freedom in a chi-squared test is number of categories of data minus 1. Whatever categories you have in total, just subtract that from 1. Now, in this case, the word categories, we didn't mention the word categories, right? So it's basically the phenotypes. How many phenotypes do you have here? Blue and black. So it's two phenotypes. So two phenotypes minus 1, that is your degrees of freedom right here, which is represented by the letter V. Simple as that. Now, I know what you must be thinking. Then why do we subtract by 2 in the t-test? We'll talk about that when we come to the t-test. So, v equals n minus 1. Sometimes this n is also represented by c, but still doesn't make a difference. Now, step number 4, and that is use the chi-squared value, use the chi-squared value and the degrees of freedom to determine the p-value using, what am I going to use to solve all of this? The chi-squared table of values. It's basically a table of values. Now, don't worry, you don't have to remember any table at all. It's going to be given to you in the question. Let's take a look at what that table is going to look like. So it's going to be a table, a rectangular shaped box. On the x-axis, or you could say in terms of what's going to be written right here, it's going to be all just p values. And then in terms of the columns, as in what's going to be written here, it's going to be the degrees of freedom. So I'm just going to write V in this case. So the P values are going to be 0.99. I'm just copying off a random table. Let's say this is going to be 0.95. Then let's say this is going to be 0.5. Then the next one's going to be 0.25, or let's say 0.05, then 0.01. Notice that not any of these p values are equidistant. They are equidistant, not at all. It's different differences between these values, so you don't need them to be equal, and plus you don't have to worry about them. You're not making them on your own. You just have to know how to be able to use them. And here you have in the columns the degrees of freedom, which is 1, and then it's much more than just 4. So 2, 3, then 4, 5, 6, and then 7. Now, how do you use this? First, understand what your degrees of freedom are. Let's say you have two phenotypes here, which you have. Subtract that by 1, and you have your degrees of freedom. So in this case, V is equal to 1, which means you're only going to be looking at this row right here. The rest of the table is not of any importance to you. Now, how do you look at this row then? You will now look at the critical value of chi squared, or the critical chi squared value. Now, what is this critical chi squared value? Simply, it is just the chi squared value when p is equal to 0.05. So, you now know your degrees of freedom, so you now exactly know which row to look at. Now locate p equals 0.05. P equals 0.05 is right here. The chi squared value corresponding to that in the degree of freedom row which you're looking at. So this right here is your critical value of chi squared. So you now have identified the critical value right here. Once you have identified the critical value, now use your value of chi squared. Now, the way this table works basically is the moment you go from left to right, meaning the moment you go in this direction, you can easily see The chi-squared value, let me write this using a stronger, yeah. You can easily see the chi-squared values to increase. Now, what do you have to do? Let's say the value here was 13.3. I'm making up random values. What value did you have? 18. Now, if you want to go to 18 from 13.5, 13.3, you're gonna have to increase, right? You're not gonna go backwards, you're gonna go forwards. How do you go forwards? By going to the right. Because when you go to the right, the chi-squared values increase, right? The only way you're gonna go from 13.3 to 18 is if you go to the right. If you go to the left, you're gonna be less than 13.3. So you have to go to the right. Notice what's happening to the p-values. The p-values are going to become less than 0.05, right? See, the p-values are decreasing. So if the p-value becomes lesser than 0.05, you now reject the null hypothesis. I know most of you would not have understood this. Let's try this again. You have your degrees of freedom. You now know which row to look at. The way a chi-square table works is if you go to the right, the chi-squared values increase. You take your value, you compare it to the critical value. If your value is greater than the critical value, of course, you have to go to the right, isn't it? And when you go to the right, you clearly see the p-values are decreasing. If you go to the left, you clearly see that the p-values are increasing, but the chi-squared values are decreasing. So it works oppositely. But in this case, we have to go to the right. Why do we have to go to the right? Because our value is greater than the critical value of chi squared. Now, while you are going to the right, you can clearly see that your p-value becomes less than 0.05. Let's say 18 would be somewhere here, and corresponding for that, the p-value would be 0.003. It's less than 0.05, right? That's what I've written here. Now, if the p-value is less than 0.05, you reject the null hypothesis. What was the null hypothesis? It was that there is no significant difference between the observed and expected values. So you reject it by saying that no, there is a significant difference between the observed and expected values. However, if let's say your value was lesser than the critical value, if let's say you got 12 as the answer, obviously you now have to go to the left, right? And while you're going to the left, you see that, oh, the p values are increasing, which means your corresponding p value is going to be more than, or even if it is equal to 0.05, it's the same thing. And what is that? If the p value is more than or equal to 0.05, you accept the null hypothesis and you say that yes, there is no significant difference between the observed and expected values. But why are we doing that? It's because you first need to understand that you need to understand what p means. P is basically the probability that the null hypothesis is true. Let's say I tell you that wherever you live, today, what's the date? 9th May. So let's say I say that on 9th May, wherever you live, there's going to be a 3% chance of strong rainfall. Just 3%. Would you tell this to another person and tell them, oh, there's going to be rainfall today? No, there's a 3% chance. It's close to zero, right? You can say it's negligible, it's just 3%. Versus if I told you there is a 75% chance of rainfall, you'd go to your sibling and you'd be like, oh, there is strong rainfall today. There's a 75% chance of it happening. So you see, 3% versus 75%. Apply this in this case. If the probability that the null hypothesis is true is less than 5% or 0.05. It's the same thing. Would you agree with the null hypothesis? You would say, nah, hell no, the null hypothesis is wrong. You reject it. Versus, if the probability that the null hypothesis is true is more than 5%, or let's say even equal to 5%, you'd be like, okay, might be cooking. You might accept the null hypothesis, and you will in this case. You will say, yes, there is no significant difference between the observed and the expected values. So that's basically how the chi-square test works.